The first speaker is Dr. James Briscoe, Group Leader in the Developmental Dynamics Laboratory at the Francis Crick Institute. Thank you.
So thank you very much. So first of all, thank you, the Allen Institute and Michael. It's a real pleasure to be here. So I wanted to actually start off with a dedication. So some of you may know Thomas Jessel and have heard that he died a couple of days ago. So Tom was my postdoc mentor and was a huge inspiration to me and to many other people. And it's particularly fitting to dedicate today to him because I know he would have really enjoyed this symposium had he been here. So as a developmental biologist, what I'm interested in is understanding how tissues make the right cells in the right place at the right time and in the right numbers. And I think of this as the 4 Ps of developmental biology, so pattern, position, precision, and proportion. So whether we're talking about a Drosophila embryo or the order of digits on your hand, or indeed the different types of neurons in your nervous system, function follows form. You need the right cells in the right place in order for the tissues to function properly. So how does this work? So how does a cell know where it is in a developing tissue, and how does it use that information to do the right thing? So we've already heard one way in which this is achieved, a common mechanism that you find in many developing tissues, this idea of a morphogen gradient. So some kind of long-range signal emanating from a localized source spreading through that tissue. And now that gradient, the concentration of that signal becomes a correlative position. If you know the concentration, you know how far you are from the source of the signal. So we've heard several talks that have thought about this in terms of how gradients are formed and how that is controlled. But I want to take you from a different perspective. So I want you to think of the perspective, thinking of yourself as a cell sitting within that gradient. So how do you see and respond to that concentration, that ligand, that signal outside of the cell? And if we think about this, this becomes a sort of an information processing problem. The cell is receiving some input, that morphogen signal, and it has to use that information to adopt the right cell identity, to express the right genes, to become the right cell fate. So how does that work? So before we get too sort of abstract and conceptual about this, let me take you back to a more concrete, specific example. And that's the vertebrate nervous system, and more particularly the spinal cord. So we started the spinal cord because we think it's interesting. But in addition, it's a simpler and experimentally tractable model that tells us things about other regions of the nervous system and indeed about other tissues. So in the spinal cord, as in other regions of the central nervous system, different types of neurons are situated in different locations. So in the dorsal spinal cord, so dorsal is closest to the skin of your back, are the neurons which receive information from the periphery. So these sensory neurons receive that information, then either send it up into the brain or send it down into the ventral region, so ventral close to your belly, so ventral regions of the spinal cord, where the motor neurons and interneurons that control motor output. So the motor neurons which send axons out of the spinal cord and innervate muscles are located, together with a set of locally projecting interneurons which help coordinate motor output. So that organization of different types of neurons arises early in development with the spatially segregated generation of these different neurons. And these neurons are generated from proliferating progenitors arrayed along the dorsal ventral axis. Now, just as we can identify different types of neurons, we can also identify different types of progenitors. And we can identify them on the basis of their gene expression profiles. And using this set and other genes, we can see that these progenitors are allocated into territories into domains based on their combination of gene expression. So each particular dorsal ventral domain is identified by the unique combination of genes it expresses. Now, experiments I'm not going to get into shows that manipulating the gene expression code by misexpressing, removing, or adding genes to this has predictable consequences on the neuronal subtype generated. So this problem of pattern formation in the spinal cord becomes one of understanding how this pattern of gene expression within progenitors arises. And this pattern of gene expression is directed by secreted signals emanating from the dorsal and ventral poles. So dorsally, signals including BMPs and WNTs are important for the pattern of gene expression, whereas ventrally, the secreted molecule sonic hedgehog emanating from the notochord, which sits underneath the spinal cord, and cells at the ventral midline are important for the pattern of gene expression in ventral progenitors and the production of the neurons which arise in the ventral region. And I'm going to focus on hedgehog signaling and the ventral neural tube for the rest of the talk. Now, several lines of evidence suggest that hedgehog signaling functions at distance, at long range, to induce these different types of neurons. So, for example, one type of experiment we can do is to explant out a little region of the forming neural tube here from chick embryos and culture that ex vivo in tissue culture and expose those explants to different concentrations of recombinant hedgehog protein. So in the absence of any hedgehog, most of the progenitors, most of the cells in these explants express a gene PAX7 characteristic of the dorsal neural tube. If we culture in the presence of a moderate concentration of hedgehog, so 1 nanomolar hedgehog, PAX7 is not expressed, and instead OLIG2, this red gene, characteristic of the middle of the ventral neural tube, is expressed. If instead of 1 nanomolar we use a higher concentration, 4 nanomolar, then you see the induction of this green gene, NKX22, characteristic of the progenitors, the very ventral neural tube just adjacent to the floor plate, the source of hedgehog, at the ventral pole. So this looks like a classic kind of morphogen, a concentration-dependent response. There's a good correlation between the concentration response and the position within the neural tube. However, of course, things are more complicated than this. So these assays are all done at one particular time point, so 24 hours after explanting. If we look at similar experiments but assayed at different time points, we see the following. So if we expose explants to that high concentration 4 nanomolar hedgehog I just showed you at 24 hours, it induces a high response gene, NKX22. But if we look at 12 hours, then the majority of cells in those explants are expressing the low response gene, OLIG2, and you see a transition to NKX22 over time. So you see initially OLIG2 being induced, and then it's turned off as the high response gene, NKX22, is induced. And indeed, if we look in vivo, we see a similar sequence of events. So this initially cells in the ventral neural tube turn on the low response gene, OLIG2, before transitioning to the high response gene, NKX22. So we want to understand how both the amount of hedgehog and the duration of hedgehog is controlling this pattern of gene expression. So how is this information being used by the cell to generate the right pattern, the right profile of gene expression? And what I'm going to argue is that crucial for this interpretation, crucial for converting the signal into the output, is the downstream response genes themselves. So as well as being nice markers for different positions along the dorsal ventral axis, all the genes I've been talking about are themselves transcription factors, and they're connected together in a transcriptional network. And that transcriptional network is crucial for this interpretation, converting level and duration of signal into patterns, these spatial patterns. And some of the evidence for this initially arose from classic developmental genetic experiments. So for example, we can now look at mice lacking one or more of these transcription factors and look at the effect of that absence on the rest of the gene expression pattern. So here we're looking at mouse embryos and two genes, NKX22, that high response gene. And you can see it's expressed in the ventral neural tube. And its expression is mutually exclusive with this green gene, PAX6, which is expressed throughout the rest of the neural tube. If we look in a mutant mouse lacking PAX6, then obviously there's no PAX6 protein. But what you can also notice is a marked expansion in NKX22 expression. Now, the dorsal limit of NKX22 is much further away from the source of hedgehog than in wild-type embryos. So this is this is a type of genetic evidence that suggests that the downstream response genes, such as NKX22 and PAX6, are themselves involved in setting their spatial patterns of expression. So from these type of experiments, we can begin to build a gene regulatory network which connects pairs of these transcription factors together. And you can notice there's lots of cross-repressive interactions. There's lots of repressive interactions within this network. So from this network, we can extract out smaller circuits comprising fewer of these transcription factors, and then begin to examine this in more detail. And I want to focus on a network composed, or a circuit composed of 4 transcription factors, 3 of which you've already seen. So PAX6, NKX22, and OLIG2. I'm going to add a 4th one, IRAQA3. And 2 of these transcription factors depend on hedgehog signaling for their expression. And then the regulatory interactions you see cartooned here are generated from the type of experiments I just showed you, these knockout experiments or gain-of-function experiments. So this set of 4 transcription factors defines 2 boundaries of gene expression within the neural tube. So NKX22 is that high-response gene expressed very ventrally in the neural tube, and its dorsal limit of expression corresponds to the ventral limit of OLIG2 and PAX6, whereas the dorsal limit of OLIG2 is coincident with the ventral limit of IRAQA3. So what we wanted to ask is, does this network explain this pattern of gene expression? And if so, how is this circuit generating this spatial pattern? So one way we've approached this question is to take a mathematical modeling approach. So we can convert this cartoon network into a set of linked ordinary differential equations which just describe those interactions in terms of rate equations. I'm not going to get into how we then parameterize this network because it's published and we don't have time right now. But having written this model and then parameterized it, we can begin to explore how this behaves, this network behaves in silico. So for example, in silico, we can now simulate a gradient of hedgehog signaling and then look at the pattern of gene expression that arises. And reassuringly, because this is what we asked the model to do, we see the expected pattern of gene expression. So a stripe of NKX22 at high levels of signaling, a stripe of OLIG2 at moderate levels of signaling, then the dorsal genes PAX6 and IRAQA3 where there's low or no hedgehog signaling. Moreover, if we simulate the genetic mutant, so for example, if we remove the PAX6 equation, then the simulation behaves appropriately. You see an expansion of NKX22 to lower levels of hedgehog signaling and a reduction in OLIG2 expression. And indeed, that's what I showed you, this expansion of NKX22 in the absence of PAX6. So this is good, this is reassuring, but this is what we asked the model to do when we established it. What was more striking is that what we noticed is that the model also explained the dynamics of gene expression. So this isn't something that we asked the model to do when we set it up, but we noticed that any set of parameters that gave us the appropriate spatial pattern of expression in simulation also gave us the appropriate dynamics of gene expression. So for example, if we simulate the model over time with a level of hedgehog signaling that at steady state gives us the high response gene NKX22, we always see a transient induction of OLIG2. So you see OLIG2 transiently being induced before NKX22 is induced and becomes stable. And if you remember, that's what we're seeing experimentally. We're seeing transient induction of OLIG2 before prior to the induction of NKX22. So what this is suggesting then is that that regulatory network, the logic of that regulatory network, is explaining both the spatial pattern of gene expression and the temporal sequence of events, the dynamics of gene expression. So we can sort of think of this in this sort of cartoon form where we're looking at the effect of the level of hedgehog signaling or the duration of hedgehog signaling. And you can see to get the high response gene, you need levels of hedgehog signaling above a certain amount. So that's kind of a classic Morphogen response. But you also need that level of signaling for a sufficient period of time. So that's the duration response. Moreover, to get from the initial conditions of IRAK3 and PAX6 expression to the high response NKX22, you always transient through a period of OLIG2 expression. The only way to turn on NKX22 is with a transient, is through a transient induction of OLIG2. So this dynamical system appears to be explaining the experimental observations. And I just want to emphasize a couple of elements of this, a couple of the features of this dynamical system that begin to explain pattern formation. So we've already had this question about how you generate a Boolean response, so how you go from an analog input, the gradient of gradually changing levels of hedgehog signaling, to these all or none changes in gene expression. And we can see this from the perspective of the dynamical system now. So if we look at, for example, the expression of OLIG2 at steady state in response to different levels of hedgehog signaling in what in dynamical systems terminology is called a bifurcation diagram, we can see below a certain level of hedgehog signaling, OLIG2 is off. But at a certain level of hedgehog signaling, OLIG2 is turned on. It's turned on suddenly. And that sudden turn on is called a bifurcation. And in fact, it's a specific type of bifurcation, a saddle node. And then OLIG2 is on until it reaches a certain threshold of hedgehog signaling, and then it's turned off, and again, that's a bifurcation, and again, it's through a saddle node bifurcation. So this dynamical system is explaining how you get those, how you convert a gradual change in hedgehog signaling into an all or none change in gene expression. And it's based, and one can intuit this through those cross-repressive interactions. Those cross-repressive interactions mean you can have one or the other gene on, but not both simultaneously. Moreover, we can use the tools from dynamical systems to think about the dynamics of gene expression as well. So if we look in what we call gene expression space, so here we're graphing the level of PAX6 from low levels of PAX6 to high levels, so that's the dorsal gene, OLIG2 from no OLIG2 to high OLIG2, and then NKX22 from low NKX22 to high NKX22. The cells start off expressing PAX6, but no OLIG2 or NKX22. If we add in the simulations a moderate level, a low level of signal, then the system moves, it downregulates PAX6 a little bit and turns on OLIG2, but NKX22 isn't expressed in those conditions. So this gives the dynamics of gene expression, simple dynamics from PAX6 to OLIG2. However, what happens if we simulate this with those higher levels of signal that result in eventually NKX22 induction? So if we do that, what we notice that instead of the system moving directly from PAX6 expression to NKX22 expression, the system initially moves towards where the OLIG2 steady state would be expected to be, but it's not there, right? So you've got a high level of signaling. The system moves towards there, but there's no steady state to find, and then falls back into, or relaxes into the NKX22 steady state. So this gives you the dynamics of the system in gene expression space. And again, in dynamical systems terminology, this behavior of moving away from before falling into the steady state is called a dynamical ghost. So there's a ghost of the OLIG2 steady state still within the system. So together that suggests an explanation for both the position and the pattern of gene expression. The regulatory logic of that transcriptional network is taking the input of hedgehog signaling and converting it into these profiles of gene expression via the regulatory links within the circuit. But what we noticed when we looked back at experimental data is that there was something else going on as well. So if we look back at the PAX6 mutant, I already showed you that in the PAX6 mutant, NKX22 expression is expanded dorsally. So compared to wild-type animals, you see the domain of NKX22 is broader. But in addition, what was obvious looking at this is that the precision of the boundary between NKX22, the green gene and the red gene, NKX22 and OLIG2, is much sharper in the wild type than in the PAX6 mutant. In the wild type, you've got a reasonably sharp boundary between OLIG2 and NKX22. But in the PAX6 mutant, now you're seeing much more intermixing of cells. So cells are still being decisive. They're mostly red or green. But there's no longer a sharp delimitation, a sharp boundary between those two domains. So what this is suggesting then is that the transcriptional network is somehow also providing precision to the network, to pattern formation. If we perturb the network, if we knock out one of the genes, we lose the precision in patterning. So we wanted to understand why that was the case. And so we reasoned that one possibility that could contribute this would be the natural fluctuations, the stochasticity, which is inevitable in gene expression. And so far, the simulations I've been showing you have all been deterministic. They don't take account of any noise. But of course, in reality, the small molecule numbers associated with gene expression processes result in random fluctuations around the steady state. So you see, over time, changes, so fluctuations in gene expression. And those when you think about the effect of those fluctuations in the context of bifurcations, this has interesting consequences. So what it means is that the noise can actually drive a transition from one steady state to another steady state. So in a deterministic system, cells would never jump from this steady state to this steady state. But when you have noise, then just a random fluctuation can take a cell across a bifurcation and force it into the other attractor, the other steady state. And the frequency, the probability of that happening depends on how close you are to that bifurcation point. The closer you are to that bifurcation, the more likely a fluctuation is to take you across that bifurcation into the other steady state. So this seemed like a possible explanation for why, or we wanted to ask whether this was a possible explanation for why precision had decreased in the PAX6 mutant. So one thing we did to begin to address this was just take the deterministic ODE model I've already shown you and just add a stochastic term to this, so using chemical Langevin equations to approximate molecular noise. So when we do this to the wild-type simulations, you can see you continue to get the NKX22 domain and a boundary between NKX22 and OLIG2, but now there's some stochastic noise in the system, so the boundary is no longer absolutely precise. So using exactly the same system, but just removing the PAX6 equation, so the same parameterization, and then simulating the PAX6 mutant, what we notice is that we continue to get the expanded NKX22 domain just as you do in the deterministic system. But what's striking is that we also saw a decrease in the precision of that boundary. So now not only was the position of the boundary changed, but the precision of the boundary was decreased compared to the wild type. So why is that? So why does just introducing stochasticity affect the precision of the boundary in the PAX6 mutant compared to the wild type? So if we come back to thinking about what's happening in gene expression space, so now we're going to look at a transition from the OLIG2 steady state to the NKX22 steady state, so high OLIG2, PAC6 moderate levels, and here high NKX2-2, no OLIG2, no PAC6. So in a deterministic simulation, cells would just sit at the OLIG2 steady state and would never reach the NKX2-2 steady state. But if we look at this in stochastic simulations, you can see that now cells are fluctuating around that steady state, and occasionally a fluctuation brings them to this purple point here, which is the transition point, the bifurcation point. And if a cell hits that transition point, then it drops into the NKX2-2 state. So this is a noise-mediated transition. So just fluctuations in gene expression occasionally result in the cells transitioning from one steady state to another. So if we look at the structure of noise, the shape of the noise in the wild-type simulations, you can see that noise results in cells fluctuating around the OLIG2 steady state. But what you can notice is that the angle of these fluctuations isn't directly towards that transition point, but it's slightly angled away from it. And it's angled away from it in the PAC6 dimension. So that means cells are fluctuating around that, but they're not fluctuating directly towards the transition point. Now, of course, in a PAC6 mutant, you completely lost that dimension. So now the fluctuations are flattened down into 2 dimensions. So the same magnitude fluctuation is now directed towards that bifurcation point, and the same magnitude is therefore more likely to reach it because you're not angling away from the OLIG2. So this suggests an explanation for how this regulatory structure is increasing the precision of the boundary. PAC6 is the fluctuations, the structure of the noise, particularly in the PAC6 dimension, is resulting in decreasing the probability of noise, resulting in a spontaneous transition from OLIG2 to NKX2-2. So this was quite a dramatic manipulation, perturbation of this system, eliminating a whole node, PAC6. And we wanted to ask whether more subtle perturbations of this network, particularly altering the strength of edges, could also affect the precision of the network. And unfortunately, we came across a way of doing this, and we were interested in understanding the regulatory elements that controlled some of the genes we were interested in particular OLIG2. And we identified a regulatory element, 33 kb upstream of OLIG2, which others had shown was important for the expression, or was sufficient to drive OLIG2 expression. And this regulatory element is accessible and binds several of the transcription factors that I introduced to you. So what we did is just delete this regulatory element in mouse embryos and look at the effect of this on gene expression in the neural tube. And so if we compare a wild-type mouse to a mouse lacking this regulatory element, so this has a deletion just of this regulatory element, what you notice if you compare OLIG2 expression between control mice and mice lacking this regulatory element is, first of all, it's obvious that the size of the OLIG2 domain is diminished, and in particular, it's lost, it's retracted towards a ventral neural tube. But also what's quite striking is we look at the precision of the boundary between NKX2-2 and OLIG2. We see now a decrease in this precision. If we compare this to the wild type where there's a reasonably sharp domain, there's increased intermixing between the red and the green cells in the absence of this regulatory element. Indeed, if we measure the precision, that is indeed the case. So what explains this? So we again return to the dynamical systems, or stochastic system, and reduce the parameters in the model which describe the binding of the transcription factors to this regulatory element. So if we compare wild-type embryos to the enhancer mutant, what was striking is that just decreasing these parameters was able to recapitulate each of the experimental observations. So you can see that the size of the OLIG2 domain is diminished, particularly the dorsal limit is now closer to the ventral neural tube. But more importantly, you can see the precision of the OLIG2 NKX2 boundary is also decreased. And that isn't because the overall noise of the system has increased. So if we look at the overall magnitude of noise, gene expression isn't more noisy by deleting this regulatory element. Instead, what's happened is the structure of the noise has changed. So if we look at simulations comparing levels of PAX6 and OLIG2 in individual simulated cells, you can see in the enhancer mutant that the prediction is that on average cells should have slightly lower levels of OLIG2 and slightly higher levels of PAX6. Then if we go and look in experimentally, actually measure levels of OLIG2 and NKX2 in individual cells, we see a similar shift in average levels of PAX6 and OLIG2. So the structure of the noise, not the magnitude of the noise, appears to have changed. So if we think about this again in the context of gene expression space, I showed you this already in the wild type, where the fluctuations are angled away from the transition point. Now, if we think about the consequences of changing the relative levels of OLIG2 and PAX6, so we're reducing slightly the levels of OLIG2, increasing slightly the levels of PAX6. If we compare this in gene expression space between wild type and enhancer mutant, you can see what that's doing is changing the structure of the fluctuations, and now they point slightly more towards the transition point. So that makes a similar magnitude fluctuation more likely to hit the transition point and result in a change in gene expression. So for the sake of time, I'm going to skip the last couple of slides. So what I want to say then in summary is that what I wanted to say is that this regulatory network explains how cells are processing the morphogen, the hedgehog morphogen input, and generating patterns that are making cells decisive, transitioning between gene expression states. And they're also positioning those boundaries and controlling the precision of those boundaries. I think this is an example where we're seeing this multi-scale behavior we often see in developing tissues. So the mechanism in individual cells, the gene regulatory network operating in individual cells, is generating a tissue-level behavior, the precision of a boundary. And this combination, coupling the experimental observations of the dynamical system, is beginning to give us insight as to why this is happening. So I'd like to finish there and just thank the people in the lab responsible for this work, so particularly Catherine Exelby and Edgar Herrera Delgado, supported by Ruben Perez, and my two long-term collaborators on this project, Karen Page and Peter Solich. And thank you very much for your attention.
We have time to take some questions. Just raise your hand and we can get a mic to you. Up and back in the middle.
So as I understand it, when you're doing these stochastic differential equations, you're just doing what they call linear noise approximation. You just add a little noise in at the end. So I guess I'm a little concerned, since you're talking about some fairly subtle aspects of the noise as which direction things are pointing in, for example, that, is it really okay to do that, or would it be better to actually put the noise in the actual generating things that generate the noise?
So I didn't get into the details of how we parameterize the noise, but we've tried to do that from experimental data. So by measuring so both molecule number, so we have some estimates of numbers of molecules, so PAX6, OLIG2, and NKX2. And we also have measurements of coefficient of variation of gene expression from experimental data as well. So we've used those to actually parameterize the CLEs, the chemical Langevin equations. So, I mean, it's our best attempt at describing the noise that we can measure experimentally.
Question down in the middle.
Early on, you showed the gene expression kinetics for each of these genes, and I was just wondering how that changes with the enhancer deletion, and whether you can tease out the timing of these events, and how much of it is due to timing versus the expression levels.
Yeah. So in fact, one of the predictions that came out of the simulations of the enhancer mutant, which was experimentally verified, is the prediction is that OLIG2 should be delayed in its induction in the enhancer mutant compared to wild type, and that is indeed the case. In terms of what is controlling the actual kinetics, the predominant parameter so in the simulations, the major parameter which controls the rate at which this system evolves is the half-life of transcription factors. So if you make the transcription factors more stable, everything slows down. And again, we have some estimates of transcription factor half-life from experimental data, which are coming out around sort of the 3-4 hour mark. And that's actually, again, in the simulations, that's consistent with the rate at which we're seeing the transients, the gene expression transients, which is on the order of 3 half-lives, so sort of the 8 to 12 hour range.
Okay, thank you so much.